Hey, do you guys also get the pop-up window that says this meeting's being live streamed, or do only I get that? Okay, everyone gets it.
Everyone gets it, yeah. I like that feature. If somebody doesn't want their video or something to be there, I was like, FYI,
yeah, it's
recorded.
It should be like clear so that someone isn't bamboozled, that they think they're being not recorded and then they are. Okay, we are live, so we've got that going, and we will just wait a couple minutes.
So I think we will go with Kolade first and then Nicoletta, because we go from early career, even though you both are early career, but still. Kolade is more
Kolade, do you have to go? You said you have something like a hard deadline, right?
No. I don't have a hard deadline. Yeah.
Okay. You were so flexible with the scheduling, which I really appreciated. So thank you again so much for that.
No, totally. I understand. It's sometimes very difficult to schedule these things, and get all the times lined up and everything. So yeah.
I am so happy there's so many people, even after ASCB.
So it sounds like ASCB, you love that meeting. Should I start going to it? I've never been.
I like it. I mean, it's also to see, I love cell biology in general, but there are so many concepts in other, let's say, organism or other kind of systems, which you can draw parallels with. So it's more for me to attend those sessions, which anyway I won't.
Okay, you use it to branch out and find something different than what you get at a more specialized. Yeah. Cool. Yeah, I saw some really good looking talks on being posted on Twitter. I think there is one right now about like the cytoskeleton and probably some migration stuff. So we definitely are splitting our audience with that, like right at this moment, unfortunately. But
that one is recorded too, so I'm sure people can watch it later. Okay, maybe we should start. It's 11:01. So thank you everyone for being here today, even though ASCB is going on. If you attend that, we are grateful that you made it here. So today our first speaker is going to be Dr. Kolade Adebowale. He obtained his PhD in chemical engineering at Stanford. His PhD work focused on how matrix viscoelasticity regulates cell migration. He's currently a postdoc in Professor Samir Mitragogi's lab at Harvard, where he's investigating macrophage tumor interactions and how macrophages traffic within tumors. Today, he's going to talk about how enhanced substrate stress relaxation promotes Filopodia-mediated cell migration. So with this, I would give the virtual stage to Kolade, and thank you so much for accepting our invitation and speaking today.
Can you hear me okay and see my screen?
Yeah.
Awesome. Yeah. Let me just go to full screen mode. Great. How does that look?
That's perfect.
Awesome. Yeah, thank you for that really kind introduction, Ankita. And I'd like to start by thanking the organizers for the opportunity to present some of our work here today. And thank you all for attending, despite ASCB going on. Today, I'm going to be talking about how enhanced substrate stress relaxation promotes Filopodia-mediated cancer cell migration. My name is Kolade Adebowale. I'm currently a postdoctoral fellow in the Mitragogi lab at Harvard, but the work that I'm going to describe today is my PhD work in the Chowdhury lab at Stanford. Cell migration is an essential biological process that's important for embryogenesis, wound healing, immune cell trafficking, and cancer metastasis. What happens during metastasis, for example, is that cancer cells break away from a primary tumor. They migrate through the ECM or extracellular matrix. They enter a blood vessel, exit that blood vessel, and then colonize a secondary site. This process of metastasis is responsible for up to 90% of cancer deaths. But one thing I hope you can appreciate from this schematic is that cell migration is an important aspect of the metastatic cascade, and that furthermore, the cell ECM interaction could play a key role during this process. So this has led many groups around the world to develop biomaterials and to understand how different properties of these biomaterials can impact cell migration. For example, some groups have looked at the role of confinement on cell migration. Others have looked at how the adhesion or stickiness of a substrate affects cell migration. Others have looked at how differences in Young's modulus or stiffness can impact cell migration. But the potential impact of viscoelasticity on cell migration is not known. But why is viscoelasticity of interest? It turns out that normal and diseased tissues are viscoelastic and they exhibit substantial viscosity. And that's shown in this graph on the left, for example, where on the y-axis is the loss modulus or the viscosity in the material as a function of the storage modulus or elasticity on the x-axis. And so I don't expect you to be able to read all the data on this graph on the left, but just to point out that there is sort of a diagonal line that goes from the bottom left to the top right. And in general, any of the data points that are clustered around this line have viscoelastic properties. So they have properties of both a fluid and a solid. And so what we find is that materials like type 1 collagen or matrigel, these are viscoelastic materials, as well as biological tissues like brain, liver, lung, and kidney. These are viscoelastic materials. So they have properties of both fluids and solids. Not only is this property implicated in normal biological tissues, but it's also found in diseased tissues as well. So here again on the y-axis on the right, we're looking at the loss modulus or viscosity in a human tissue as a function of the stiffness on the x-axis. And what this data shows is that for malignant human breast tumor samples shown by the red squares, these materials are not only stiffer, so they're farther along on the x-axis in general, but they also have a higher viscous property, which means they're farther along on the y-axis. This increase in both stiffness and loss modulus has been found for not only breast cancer, but also bladder cancer, liver cancer, and pancreatic cancer. One of the consequences of viscoelasticity is that these materials exhibit stress relaxation. And what I mean by stress relaxation can be demonstrated by what we call a stress relaxation test, where we input a constant strain into a material and then measure the stress response. And here's a schematic view of what the results from that experiment look like. So here we're looking at the normalized stress of the material on the y-axis as a function of time on the x-axis. For a purely elastic material, there's no change in the stress of the material as a function of time. In contrast, however, for a viscoelastic material, the stress in the material decreases as a function of time. You can think of an elastic material like a rubber band, for example, and then you can think of a viscoelastic material like a solid material like Jello, for example, that can kind of wiggle around. Lamellipodia have been described as a universal mediator of 2D cell migration. And just to quickly remind you, Lamellipodia are these structures found at the fronts of a cell that's migrating. They are characterized by a dendritic actin network, cable-like actin stress fibers, and a very spread morphology. And we currently have a textbook view of the critical role that Lamellipodia play during cell migration. And that's, for example, highlighted by this paragraph on the bottom that says that cells that are not able to make normal Lamellipodia crawl poorly and tend not to metastasize. Over the last several decades, there's been intense focus in the biomaterials community to try to understand the potential impacts of Young's modulus or stiffness on cell migration. What's been found is that when cells are on a compliant substrate, the cells are very rounded and non-motile. But if you put those same cells on a more rigid substrate, they acquire a motile phenotype shown by this light blue region. And that associated with this transformation is a formation of Lamellipodia structures at the fronts of the migrating cell, and this actually mediates cell migration. Of note, most of these studies use very rigid or purely elastic substrates. But we would like to utilize and develop hydrogels that have independently tunable stress relaxation properties. This hydrogel consists of two main components. The first is basement membrane matrix or matrigel, shown by the green line. And the second component is alginate, shown by the dark blue line. the alginate is ionically cross-linked by adding calcium ions. And the way that we can modulate the stress relaxation properties is simply by changing the molecular weight of the alginates. That then allows us to make a range of viscoelastic materials that all have the same initial Young's modulus of about 1.5 kilopascal, or a purely elastic polyacrylamide hydrogel that has the same initial Young's modulus. These materials are quite compliant. To give you a sense of how compliant they are, a gummy bear has a Young's modulus of about 70 kilopascal. So these materials are about an order of magnitude more compliant than a gummy bear. In any case, however, we can quantify the differences in stress relaxation properties by looking at this stress relaxation curve that I introduced earlier. Again, here we're looking at the normalized stress in the material as a function of time on the x-axis. And one way to quantify the differences in stress relaxation is by looking at the time that it takes for the peak stress of the material to reduce to 1/2 its initial value. And that number is about 100 seconds for what we call a fast relaxing hydrogel, and it's about 2,000 seconds for what we call a slow stress relaxing hydrogel. So to establish some nomenclature for the rest of this talk, whenever you hear fast or slow, that is going to refer to fast or slow stress relaxing hydrogel. We like this material system because it allows us to tune the stress relaxation properties while maintaining a similar Young's modulus, constant matrix composition, and very similar pore size. This is important because later on, if we observe any differences in cell migration, we can attribute those differences uniquely to differences in substrate stress relaxation. So now we would like to understand what is the impact of differences in substrate stress relaxation on 2D cell migration. So we can make these substrates and then, for example, put HT1080 cancer cells on top of these substrates and watch how they crawl around. So here we see a cell that's moving from left to right in this video. We can then plot the migration trajectory for cells on either a fast relaxing or slow relaxing substrates. So each of these lines you're looking at is the migration trajectory of an individually cell that's migrating. And then we plotted all these migration trajectories on the same origin for ease of visualization. And there's a scale bar on the bottom left of 100 microns to give you a sense of how the cells are moving on these substrates, how far they're moving. And what I hope you can at least appreciate qualitatively is that the cells are more migratory on a fast relaxing compared to a slow relaxing substrates. Furthermore, if we quantify these differences, for example, by looking at the cell migration speed here on the y-axis, what we find is that the cell migration speed is greater when the cells are on a fast relaxing compared to a slow relaxing substrates. We've obtained similar results with MDA, MB231, and MCF10A cells. So these results are not cell line specific. We also find that fast substrate stress relaxation potentiates superdiffusive migration. And we found this by looking at the mean square displacement, or MSD, plotted here on the y-axis as a function of time. This MSD gives us a sense of how efficient the cell is migrating. And what we find is that the mean square displacement is about 2 orders of magnitude greater when the cell is on a fast relaxing compared to a slow relaxing substrate. It also turns out that this MSD metric can tell us something about the underlying statistics of the cell migration behavior by looking at what's called this alpha value. And when this alpha value is equal to 1, we have a purely diffusive or random cell migration. When it's lower than 1, we have a subdiffusive cell migration. When it's greater than 1, we have a superdiffusive cell migration. And so what this data shows is that the cells have a superdiffusive migration when they're on a fast relaxing substrate compared to a slow relaxing substrate where they have a subdiffusive migration phenotype. So now we'd like to understand what mediates the observed migration phenotype. And in the interest of time, I'm just going to focus on the results from the fast relaxing substrates because that's where we observe the most robust migration phenotype. So one of the things we observed was when we looked very closely at the cell membrane, we found these finger-like extensions protruding from the cell membrane. And these protrusions contain actin, so that's what's stained for in this image. We observe these protrusions when the cells are on a fast relaxing substrates, as well as on a slow relaxing substrates. In contrast, however, when the same cells are on the glass substrates, we begin to see evidence of these lamellipodia structures. So together, this data suggests that lamellipodia are not observed on stress relaxant substrates, but we observe a different kind of actin structure. We then wondered if these finger-like extensions have anything at all to do with cell migration, and it turns out that they do. So here we're looking at a cell that's migrating from left to right, and as the cell is migrating, you can see these finger-like extensions that form at the front of the cell that's migrating. And these protrusions are highlighted in these still images over here. It turns out that the number of these protrusions is also mechanosensitive, such that there are more of these protrusions when the cell is on a fast relaxing compared to a slow relaxing substrates. These protrusions reminded us of Filopodia protrusions. So we then stained these protrusions for myosin 10, which is a key marker of Filopodia. And we find that indeed, these protrusions contain myosin 10 protein. Not only do they contain myosin 10 protein, they contain additional Filopodia proteins such as VASP and P130 Cas. So together, this data suggests that the protrusions we observe are Filopodia protrusions. To further confirm the role of Filopodia protrusions in cell migration, we use the combination of small molecule inhibition and genetic knockdowns to perturb the activity of key proteins involved in Filopodia formation, and then ask the question, what is the potential impact of doing those perturbation experiments on cell migration, for example, by looking at the cell migration speed? And what we find is that if we use small molecules, so for example, inhibits the activity of forming or fascin, these are two key proteins involved in Filopodia formation, we find that the ability of the cells to migrate is significantly diminished. If we do a more targeted experiment where we knock down fascin-1 or myosin 10 by using siRNA, again, these are two key proteins involved in Filopodia formation, we again find that the ability of the cells to migrate is significantly diminished. So together, this data suggests that Filopodia protrusions and associated proteins play a key role where the cells are migrating on the fast relaxing substrates. We then wondered, how is it that the cells are interacting with these substrates and what kinds of adhesions are they forming? So here we use phosphorylated paxillin as a proxy for cell substrate adhesion. And we focused in on these dot-like puncta at the cell periphery, which we call nascent adhesions. We find these nascent adhesions on both the fast relaxing and slow relaxing substrates, but we also find these adhesion clumps when the cells are on the slow relaxing substrates that potentially suggest that these adhesions are somehow being misregulated through poorly understood mechanisms. We can quantify these adhesions, for example, by looking at their number and their area, and it turns out that they're also mechanosensitive, such that there are more of these nascent adhesions when the cells are on a fast relaxing compared to a slow relaxing substrates. Interestingly, however, the area of these adhesions is very similar between both stress relaxing substrates. To put this into perspective, we can compare what we would see if we had the same cells on a glass substrates. And here, not only do we see these nascent adhesions, but we begin to see evidence of mature focal adhesions that have a more elongated morphology. Again, we can quantify these adhesions, and there's dramatically greater number of these adhesions when the cell is on a glass substrate compared to the stress relaxing substrates. And the area of these nascent adhesions is very similar across all substrates. So to put it all together, this data suggests that for the stress relaxing substrates, fast stress relaxation promotes nascent adhesions, and that in general, cells form very weak adhesions when they're interacting with these compliant stress relaxing substrates. Next, we wondered, how is it that the cells are able to generate traction forces to support cell migration? So here we did a traction force microscopy type experiment where we embedded fiducial markers, which are shown by these red dots, inside the hydrogel, and then looked at how these beads or fiducial markers are being displaced in response to a cell-generated motion. So here this cell is shown by the green picture. And if you focus in on these yellow squares for a second, what I hope you'll be able to see is that in response to cell migration, the beads are actually being displaced. So the matrix is being displaced. We can visualize this displacement by these heat maps, for example, and the heat map is color-coded such that the cooler color indicates a low substrate displacement, and the warmer colors indicate a high substrate displacement. And so here we're looking at how the substrate is being displaced as a function of time. And the arrows give you a sense of the direction in which the substrate is being displaced. In addition, we can quantify the maximum traction strains that are being generated when the cells are migrating on either a fast relaxing or slow relaxing substrates. And what we find is the maximum traction stresses are much greater when the cell is on a fast relaxing compared to a slow relaxing substrate. Furthermore, we find that cell migration is impaired if the canonical traction force generation machinery is inhibited. For example, if we partially inhibit actin polymerization, or if we partially inhibit myosin 2 activity, or if we use blocking antibodies to partially block beta-1 integrins, either of those perturbations significantly reduces the cell migration speed as shown in these images. To obtain additional molecular insight into what's going on, we wanted to implement this motor clutch model of cell migration. And this is work that we did with our collaborators at Penn and Minnesota, Professor Vivek Shenoy, Dr. Zha Gong, Professor David Odie, and Dr. Jay Hu. And this model consists of three main components. The first is actin polymerizes at the front of the cell that's migrating, and this creates a protrusion and forms an actin filament. The second component is that there are myosin proteins that bind to that actin filament and pull that filament toward the cell body. And the third component is that there are clutch molecules, such as integrins, that the cell can use to interact with these stress relaxing substrates. So the first thing we wanted our collaborators to do was to be able to reproduce some of the basic migration phenotype that we observed experimentally. And we were really excited when the simulation showed that the mean square displacement is much greater when the cells are on a fast relaxing compared to a slow relaxing substrates. So these simulation results match very nicely the trends that we observed experimentally, and this gave us a lot of confidence in the model. We then wanted this model to be able to make some additional predictions that we can actually test in the lab experimentally. So here what I'm highlighting with the red arrows are Filopodia protrusions. And as I play this movie, hopefully you can see that these protrusions have some kind of dynamic behavior, so they can extend and they can retract over time. And so what the simulations predicted is that these Filopodia protrusions are mechanosensitive, such that the length of these protrusions and the lifetime of these protrusions are both greater when the cell is on a fast relaxing compared to a slow relaxing substrates. We were then really excited when we went into the lab to test these simulation predictions. And what we found experimentally is that both the length of the protrusions as well as the Filopodia lifetime is both greater when the cells are on a fast relaxing compared to a slow relaxing substrates. So these experimental results matched very nicely what the simulations predicted. To put this all together, we propose a model where we have a cell that's sitting on top of a stress relaxing substrates. And for now, I'm just going to focus on this interface between the cell and the substrates. So this interface consists of three main components. The first is this green rod, which represents actin filaments. The second component is the substrate, which is shown by this brown rectangle. And the third component is that there's this spring, such as integrins, for example, that connects the inside of the cell, the cytoskeleton, to the outside world, which is the brown rectangle. And this green rod is being pulled to the left by myosin proteins, as I mentioned earlier. So because these materials are viscoelastic and they have stress relaxation properties, when you tug on them, when you pull or push on them, the Young's modulus actually changes. So the initial Young's modulus of the substrate is color-coded such that the darker colors is the initial Young's modulus. And then as you pull and push on this material, the Young's modulus actually changes to this lighter brown color, which is the long-term Young's modulus, and it's usually lower than the initial Young's modulus. But at the same time, we have this mechanical connection. This rod is being pulled to the left, and as it's being pulled to the left, it's connected to this spring, and these springs become stretched out. So the integrins become stretched out, and these springs are also color-coded such that the cooler colors indicates a low bond force, and the warmer colors indicates a high bond force. So these springs get stretched out. So the color changes from blue to red. They begin to build up load, further build up load. But again, because this is the stochastic process, eventually this mechanical connection is disrupted, but then this cycle can repeat itself over and over again. So this is what happens when the cell is interacting with the fast relaxing substrates. In contrast, however, when the cell is interacting with a slow relaxing substrate, we find that these bonds are loading up very quickly and they're failing very quickly. So this mechanical connection is being prematurely disrupted. And perhaps one way to think about this is that when you think of the slow relaxing substrates, it's similar to pulling on a car strut. And so because this material doesn't have a lot of give, the bonds are loading up very quickly, but they're also failing very quickly. In contrast, however, in the case of the fast relaxing substrates, you can think of it as being similar to pulling on a slinky. And so because you're pulling on a slinky, it takes time to build load on a slinky, but that time then allows you to begin to form Filopodia protrusions, mature these protrusions, and recruit all the machinery that's necessary to support cell migration. So to summarize, what I've tried to describe is a material system that we've developed that allows us to tune the stress relaxation properties. And we can quantify differences in substrate stress relaxation by doing what I call a stress relaxation test. And then we can put cancer cells on top of these materials and watch how they migrate over time. And what I've described to you is that we have a canonical view that cells are not migratory on a compliant substrates. But in the current work, what we demonstrated is that even though the cells are non-motile on these compliant substrates, if that same compliant substrate has sufficient stress relaxation properties, the cell can transition to a motile phenotype shown by this light blue region, and that associated with this transition is a formation of Filopodia protrusions at the front of the cell that's migrating. And with that, I would like to thank Ovi for his mentorship during my PhD work, all the members of the Chowdhury family. I would like to thank our collaborators. I'd like to thank our funding sources, and this is my Twitter handle. And if anyone wants to email me, my email is kaa@g.harvard.edu. Again, if we don't get to your questions, please feel free to email me at kaa@g.harvard.edu. And thank you for listening. I'll be happy to take any questions.
Thank you so much, Kolade. That was such a clear and wonderful talk. I almost felt like you were giving a job talk and I would have given you the job. So congratulations on that, even though I have no authority to give you a job.
Thanks.
Okay, so I think we already have questions starting to trickle down. Maybe I can start with mine since I already have it and I can. So I was just wondering, with the Filopodia lifetime being longer on the fast relaxing substrate, why are focal adhesions smaller when it's taking, the load? Because I would think that it gives it long enough time to form more mature focal adhesions.
Yeah, so the question is, why are the...
So on a fast relaxing substrate, the Filopodia lifetime is longer, but the focal adhesion is smaller, right?
Yeah, so we mostly focused in on the nascent adhesions. So the size, so you said smaller, so the size is actually very similar on the stress relaxing substrate.
Got it. Okay.
Yeah.
So it's just that the number of nascent adhesions increases, but the size. But it's still interesting that they don't get mature. Maybe it's because of the way the substrate is. It's not stiff enough like a glass would be, right?
That's exactly right. That's exactly right. That's our thinking as well.
Okay, thank you. The first question I'm going to take is from Thomas Schmidt. Is the characteristic time scale fast versus slow related to the speed of the cell movements?
Oh, that's a great question. So this is referring to the T1 half that I talked about, this characteristic time scale. So we don't have any strong correlation. The only thing we can say is that for the fast relaxing substrate, which is the blue line, the T1 half is about 100 seconds versus 2000 seconds for the green line. And the cells are more migratory when they're on the blue line compared to the green line. Yep. I don't know if that answers that question, but that's the relationship that we can kind of that the data shows at this point. That yeah, so the lower the characteristic time scale, the more migratory the cancer cells are on that substrate. Yeah.
Got it.
Great, thanks. We have a next question from Yutaka Matsubashi, who says, very interesting talk, thank you, agreed. And he says, if the stress relaxation is also occurring in vivo, what do you think is the mechanism? Perhaps proteolysis?
Yeah, thanks for that question, Yutaka. So the mechanism, it could be proteolysis, but there are other mechanisms that could be at work. So we have another paper that came out in 2018 that actually showed that when cells are migrating in a three-dimensional context in a stress relaxant substrate or viscoelastic substrate, they can actually migrate in a protease independent manner. And so in vivo, for example, there are different ways this can happen. For example, there are a lot of proteins in vivo that are in a sort of a they're in a confined conformation, and then if stresses are applied on them, they can sort of stretch out. So stretching out of proteins in vivo can be a mechanism of stress relaxation. Another potential mechanism is that there are a lot of cross-links in vivo that are weakly cross-linked, so they're not necessarily covalently cross-linked. So again, because they're weak, if there's any kind of mechanical perturbation, those cross-links can actually come off, and so that could be another potential mechanism in vivo. Yeah.
Thank you very much. Great talk.
Thank you. So the next question is from Luca Struowski. Says, thank you for your talk. On the movie that you showed, slide 12, probably, the cell moving on the fast relaxing gel seemed to send out most protrusions while it stopped mid-run. Is this observation true? Would it be then the means to search the guidance cue?
That is exactly correct. So this is, I believe, the slide. So here we're talking about the bottom left slide. So that's exactly right. So basically, what we find is that when the cell is migrating, it's actually not a continuous process. So the cell will move, it will stop, it will kind of probe its environments, and then it will move again, and then stop, and then probe its environments. So we think that these Filopodia protrusions they're playing a role in not just migration, but also helping the cell to kind of fill out its surrounding and kind of know what direction to go and what's around. So yeah, but also, yeah, so that's what we observe. And yes, the cells are not always continuously migrating. They always have to sort of take a break after a certain time, reassess what the environment is, and then make a decision. They make a calculation. Am I going to continue moving? Am I going to go in the same direction? Am I going to change direction? So yeah. Yeah, thank you for the question.
Fantastic. I do have a question of my own, but I'm just going to ask one last one and then we'll move on in the interest of time. A technical question from Sang-Yoon Han mentions that TFM needs a reference bead image. And so in your case, where you have a gel that's viscoelastic, it should be impossible to come back to the original configuration. So how do you handle that?
Yeah, so the way that we, so this is work that we're following up on with Christian Frank's group at University of Wisconsin. We're trying to get some more accurate estimates of the traction forces. So the way that we, for example, did these, so these are, I just want to stress that these are estimates on the bottom left. So we've been very careful to use the word maximum traction stresses. And the way that we compute these numbers are to look at what the beads are doing before cell migration. And so that's our reference image. And then look at what the beads are doing after cell migration, and then compare those two things. So that's the way that we have estimated these maximum traction stresses. And again, we are following up on this with Christian Frank's group, trying to develop more realistic and accurate models for viscoelastic materials. Yeah.
Great, fantastic. Thank you so much, Kolade. That was an exceptionally clear and really fantastic presentation. All right, so now I'll invite Nicoletta to put up her slides. And we are next delighted to welcome Dr. Nicoletta Petridou. So Nicoletta received her bachelor's and also her PhD degrees from the University of Cyprus. And then in 2016, she joined the lab of Carl Philipp Heisenberg at IST Austria as an EMBO postdoctoral fellow. There, she worked on the role of tissue rheological properties in early zebrafish development. And then in December 2020, so just 1 year ago, so happy birthday to her lab, she started her own research group at the Developmental Biology Unit at the EMBL in Heidelberg. And her group focuses on experimental identification and understanding of critical phenomena and phase transitions during embryo morphogenesis. The floor is yours, Nicoletta. Thank you so much for being here and presenting your work.
Thank you so much for the invitation. It's a really great pleasure to attend the seminar series. Okay, so yeah, today I will discuss not about exactly cell migration, but about a topic that of course is very relevant to cell migration, which is material properties, as we have heard extensively in the series. But here I will talk about tissue scale material properties and how they change during development through the phenomenon of phase transitions. So the first half of the talk will be focused on a biophysical framework that it can be used to quantitatively identify material phase transitions in embryonic tissues. And this is published work. It was done during my postdoc in the Heisenberg group at IST Austria, and it was a collaborative work with theoretical physicists Edward Canezo and Bernard Corominas Murtra. Now, in the second half of the talk, I will present our very first results from my group at EMBL Heidelberg. So that where we build on this framework to address now the role of these collective material states in the embryogenic process of patterning. And this is very new, and we will really appreciate your feedback. Okay, so during tissue morphogenesis, we observe a remarkable interchange between stability and dynamism. So sometimes tissues maintain their structure and architecture, and some other times they undergo these very dramatic changes, as you can see during the gastrulating embryo. And of course, biochemistry and the mechanical forces are considered the active regulators of tissue morphogenesis, but also several lines of evidence, especially the last few years, suggest that, let's say, the passive material properties are also key in spatiotemporally controlling tissue shaping. And as demonstrated decades ago, of course, tissues exhibit characteristics of solids and fluids, meaning that if tissues have a mechanism by which they can actively change between solid and fluid states, even under maintaining like a constant force, this would be sufficient to impact their shaping. And tissue viscoelasticity, similar to cell viscoelasticity, and as we heard before, can be measured through these force relaxation tests, where a constant force is exerted and then the deformation of a tissue can be monitored. And in general, what happens to a tissue at short time scales, it deforms shortly and irreversibly like an elastic solid. And when the force is removed, it returns to its initial shape. So a solid-like tissue is expected to maintain structure and be robust against mechanical perturbations. Now, at longer time scales, the tissue increases its deformation like a viscous fluid. And when the force is removed, it acquires a new shape now. And this is an irreversible deformation. Thus, a fluid-like tissue, for example, in an embryo, is expected to permanently change shapes and thus be adaptable to mechanical inputs. Now, the spatial and/or temporal interchange between fluid-like and solid-like tissue material states was shown through many labs in excellent studies recently to guide several tissue morphogenetic processes from spreading, elongation, and wound healing. But the question that we asked here in this project in my postdoc was, how do tissues change their material properties? So what kind of cellular characteristics trigger material phase transitions? And I think this is not a trivial problem, because we need to understand how a collective macroscopic property is changing in response to some local, presumably microscopic cellular property. And as we all know, linking scales, especially in biology, is not an easy task. And here we have the help from frameworks from statistical physics that have been used to quantitatively predict macroscopic material states from microscopic interactions. And I'm giving like an intuitive example, but it's not directly related to tissues, but just the concept is very relevant. That if we think like an ice cube, which is like in the solid state, the molecules are dense and they move in order. So if we can define a macroscopic order parameter, this would be the density of the system. However, this can dramatically change as a function of some control parameter, in this case, the temperature, which when reaching what is called a critical point, it can trigger a sharp decrease in the density And the molecules now are causing this disordered molecular motion because they're just not dense anymore. So here what is important is that the molecules are still the same, but only their interactions have changed, and this is sufficient to lead to the emergence of the liquid form. And with this inspiration, such frameworks have been widely used to identify the phase space where tissue collective material states emerge. And I think the most famous example is presumably the jamming transition that is observed widely in epithelia and is in collective migration, where the tissues suddenly shift their dynamics from this freely moving and remodeling state that is more like a liquid to this frozen and static state. And there are a series of beautiful theoretical studies that provided an extensive phase space of the jamming transitions, where now, instead of talking about temperature, we have 3 control parameters in place. So jamming is expected to happen when cells either reach critical density, or they stop, move, or fluctuate, or maybe due to some geometrical constraints, such as their shape, that is through balancing adhesion and contractility, and this can limit their rearrangement. And in this case of jamming, what is expected from the tissue's perspective is that when it freezes, that its elastic modulus of the tissue will sharply increase. However, how do we go from these expectations to proving that a phase transition is occurring in the living tissues? And now this requires a combination of theoretical and empirical data, which goes back and forth, but on all scales, so at the macroscopic and microscopic scale. And this is something that we attempted to do the last couple of years using as a system the Zebrafish early embryo. And the reason why we use this system is because actually we have previously shown, and this is an older published work, that its morphogenesis, so the shaping of the embryo, relies on spatial and temporal changes in the tissue material properties. So here we see the zebrafish blastula. It is composed of the blastoderm that sits on top of the yolk cell. So this is like a multi-layered and loosely attached tissue that starts to spread on top of this yolk. And during this process, it forms this dome shape, and that's why it's called doming. Now, at this stage, what we see is that this central blastoderm deforms extensively, whereas this marginal area is resistant to deformation. And this we were able to quantitatively address by measuring the blastoderm viscosity in these regions by performing the force relaxation tests. So what we did is to use a small microneedle, a glass needle, to enter the blastoderm in the center or in the margin and apply through a microfluidic device the same negative pressure, constant pressure. And with this, we expected that the most deformable tissue will flow faster in the pipette than the less deformable tissue. And indeed, we observed that the central blastoderm displays a dramatically larger and faster deformation when compared to the marginal blastoderm. Now, from the slope of this deformation, we can calculate the viscosity of the tissues. And what we see is that the central blastoderm has around 10 times less viscosity than the marginal blastoderm. Now, if we go back in time and address how this spatial rheological difference emerges, and now it will focus only on this central area. So what we did was to take viscosity measurements every 10 minutes. And what we observed was actually very interesting, and was that the initial blastoderm displays high viscosity. And this actually is stable throughout its development for around 1 hour. And then it fluidizes very quickly, just within a few minutes at the onset of doming. And this is very transient. And then it starts to recover these viscous properties gradually through this slow thickening process. So we'll have a very abrupt nature of blastoderm fluidization that mimics this idea of phase transitions, that when you go from solid to fluid and vice versa. So it was a good point to start asking if this tissue undergoes a material phase transition. And to address this, of course, the first thing to do was to go to the literature and explore these key jamming parameters, and then see if there are any correlations of these parameters with our viscosity measurements. And quickly we were quite disappointed because we didn't find any correlation between the two. So perhaps we were looking here in this system in the wrong parameter. And then it was actually much simpler than what we thought. And this started to clear up when we start looking on the parameter of cell connectivity. And what I mean by this, so we took confocal sections of the zebrafish blastoderm that is labeled for nuclei, membrane, and interstitial space. And what we consider is that, okay, if two cells appear to touch each other, we consider that they have a contact, but if they don't appear to touch each other and there is interstitial space between them, then they don't have a contact. And in this way, we build connectivity maps, so binary maps, from which we can calculate the average connectivity of the tissue. And this analysis revealed here now a clear nonlinear relationship between tissue viscosity and blastoderm connectivity. And what was very surprising to us is that tissues that have very similar connectivity appear to have dramatic differences in their viscosity. One example is at the blastula stage, where the blastoderm, as I told you, displays 10 times more viscosity than the blastoderm at the dome stage. However, their difference in connectivity is very small. It's only around half a contact per cell. So we have a pattern of smooth changes in a control parameter, connectivity, that leads to big changes in the order parameter in the viscosity. So this resembles a phase transition. So the question that we wanted to address is what kind of phase transitions? And here this is when we team up with Bernat and Eduard. And when they saw this plot, what they noticed is that if we assume that the cells have a maximum connectivity around 6 contacts per cell, which is kind of the stereotypical cell packing, this potential transition point corresponds to the two-thirds of this maximum potential, of this maximum connectivity. And now this value, specifically the 2/3 of the maximum connectivity in networks, so in random networks, corresponds to what is called the critical point of a rigidity percolation transition. And by rigidity, what we mean here is a structural configuration that it cannot be deformed unless there is an expense of energy. So if we look at a very simple motif like this, so let's say it has 4 nodes with 4 links. If we try to pull this motif, it will deform. However, if we just add 1 contact that will absorb the degrees of freedoms of these 2 nodes under the same deformation force, it will not be deformed. And this is what we consider rigid. Now, what matters at the rigidity of a macroscopic structure like in the network is not how many rigid clusters we have, but what is the size of the biggest rigid cluster in the network that we call giant cluster, and you see in this plot in red. So it was predicted actually for a long time ago that when the connectivity of a random network is just above the 2/3 of its maximum potential connectivity, there is a dramatic structural change in the network. And this is the nonlinear emergence and increase of the size of the giant cluster. So you see here in red in the network, it almost occupies the whole network. So the whole network is rigid. However, just having connectivity that is just below this critical point, the giant cluster abruptly disappears, and that's why the network is floppy. So the question is, can we find these characteristics of the giant cluster in vivo? And when we look for it in the embryo, so you can see here the connectivity maps on the left and the rigidity maps for the giant cluster on the right, we see that it is formed before morphogenesis, so at these blastula stages, but at the onset of doming, so here it abruptly disappears. And then slowly we see that it starts emerging again during this thickening process where the blastoderm recovers its viscosity. So there is a temporal evolution of the giant cluster that seems to match the temporal evolution of tissue viscosity. We can also see that all of our data points fall very nicely on this master curve of the emergence of the giant cluster. But what was very important to us is that when we compare this experimentally measured viscosity of the blastoderm with the experimentally measured fraction of this giant cluster, we see that embryos with big giant cluster are indeed rigid, where embryos with small giant cluster are fluidized. And importantly, the fluidized embryos display connectivity that is below this theoretically predicted critical point of the rigidity percolation theory. And here I will need to cut the long story short, just to tell you that we have applied this theory together with experimental measurements in a series of embryonic tissues from different genetic backgrounds, positions, mutants, to explore how universal is this connectivity critical point. So we had around 900 networks, more than 1,000 viscosity measurements, and we do observe that the connectivity is a simple but very robust predictor of tissue rigidity in a parameter-free model. So now this next step for us was to understand the cellular biology behind connectivity. And this is a question that we always get. So connectivity, what does it mean? Cells are active, so just by appearing to touch each other doesn't mean much. And of course, it is intuitive that cell connectivity would be regulated by cell-cell adhesion, but how exactly? So how much one needs to change the cell adhesion to trigger the assembly or disassembly of one contact? So to address this, we went even more microscopically and explored a bit the biophysics of the cell-cell contacts and how they can influence connectivity. So the strength of the cell-cell contact was previously shown in a work from Takashi Hiragi group that is defined by the ratio of the cell-cell tension, so that is between two cells, and the cell cortex or fluid tension, so at the cortex of the cells. And this can be calculated by the angle that is formed at the contact edge between two cells. So the bigger the angle, the smaller the cell-cell tension alpha, and thus the stronger the adhesion. So it's a bit of an opposite relationship. So we perform simulations on simple cell motifs where we change the angle and address effects on their connectivity. And as you can see here, we identify a small range of alpha that the cells suddenly increase their connectivity. So they just add one more contact and then the whole motif becomes rigid. But this is also true for networks, so for global scales in simulated tissues, because we could also detect that this was conserved where networks with high cell-cell tension, so small angles, are floppy, whereas networks with low cell-cell tension and bigger angles, you see that it is just by changing the angles is sufficient to get them more connected and form these large giant clusters. However, these alpha values, are they relevant to our in vivo system? So we checked these values in vivo, and we observed that indeed at blastula stage, the cells are more adhesive, and they have bigger angles, whereas at the fluidization stage, the cells are less adhesive and they have higher alpha. And we also perform similar manipulations as those that I've shown before, so different mutant embryos, and we see a very nice conserved relationship with high cell-cell tension alpha being less connected and fluidized. So this now brings me to the second part of my talk, which is now that we have a more complete link between this microscopic adhesion, mesoscopic connectivity, and macroscopic rigidity. Can we explore this function of collective material states in development? Which, of course, yes, on the one hand, we expect that it defines the deformability, so the mechanical state of the tissue, but beyond that, we expect that it will also receive and presumably feed back to biochemical and mechanical signals of its environment. And this is a question that Camila, so she's one of my first PhD students in the lab, that she started addressing at the beginning of this year. So what she did is she went back to the zebrafish dome stage. That's, as I showed you before, where we just zoom either in the central or in the marginal regions. We have this pattern of central fluidization in the center and marginal rigidity. And here she also measured the cell-cell tension alpha and is in agreement with what we had so far. Now, in principle, these networks are isotropic when looking at them in isolation, but what happens if we zoom out? Because there is this specific time window that they coincide, suggesting that if we look at a larger scale, at some point in space, an anisotropy may emerge in some of these features, either the cell-cell tension, the connectivity, or the rigidity. And this is one of the first experiments that Camila did. So she imaged this process in a lower magnification, but still high resolution, and selected a larger area that includes now the previous marginal area, but extends further towards the center of the embryo. And then she performed rigidity percolation analysis over time. And she observed first that now there is a geometrical bias where this giant cluster will appear. So it's only at the marginal side of the embryo. And second, that there is a sharp material boundary that is perpendicular to the animal vegetal axis of the embryo, that at least it seems that mechanically it excludes this marginal area from the rest of the embryo. So once we saw that, the first thing we ask is how the control parameter, so the connectivity, behaves in space. And to do this, Camila separated the network in 4 regions of interest, so ROIs that are parallel to the margin, and measured connectivity in each ROI. So here I just turned this 90 degrees to match the plots. And so here we have the vegetal pole to the left and the animal pole to the right. And as you see here, this analysis actually revealed that there is a gradient of cell-cell connectivity along the animal-vegetal axis with high connectivity at the margin and low connectivity towards the center of the embryo. And given now that this gradient spans connectivity values that are above and below the critical point, this rigidity phase transition now happens in space. Now, this suggests that the range of the gradient may affect the geometrical bias where this giant cluster appears. And this we started exploring by going back to our simulated networks and now introducing anisotropy, so a gradient in connectivity. So now what we notice is when we introduce a steep gradient, we'll have a very well-defined boundary emerging. However, when we introduce a shallow gradient, we have a noisy and imprecise material boundary emerging. So these predictions imply that a gradient of cell-cell connectivity is patterned along the animal-vegetal axis, and its steepness may regulate the precision of the mechanical boundary formation between the marginal area and the rest of the embryo. This gradient is also evidenced at the cell-cell adhesion level, where we can measure the cell-cell contact angles, and we observe that they become smaller towards the animal pole, and thus the cell-cell tension becomes bigger. And this we can monitor over time, so this gradient becomes more and more rigid, more and more defined. So what might be the role of this gradient and the resultant material boundary? So during the time window that the mechanical boundary is formed, the specification and the fate of the marginal zone of the embryo kicks in. So these areas, this marginal area acquires the mesoendodermal character. So we can image it in transgenic embryos that express GFP under the promoter of CPOX. This is an early mesoendodermal gene, whereas the rest of the embryo will eventually become the ectoderm. And now segregating the two layers is key at this stage, since the mesodermal cells are those that will after internalize and migrate during gastrulation and build the body plan. And so far, no physical boundary was found to separate early on these two layers. So here we hypothesize that these material differences between the two populations may be important for precisely segregating them. And what we see is that in wild-type embryos, where they build this very nice adhesion gradient, the specification zone is very well defined. So we can evaluate this by the signal distribution of CPOX along the animal-vegetal axis. And this specified region is nicely encompassed within the rigid material. So as you can see, we have a sharp decrease of the giant cluster along this axis. It coincides in space and time with the sharp separation of the biochemical differences in the fate of the tissue. So we have a nice correlation how we can explore a functional relationship between rigidity and germ layer segregation. So we don't have the answer yet. I'm just showing some hints that they point towards this direction. So first we ask what sets up this adhesion gradient, because if we can change the gradient, we can then address how this emergent rigidity can affect cell fate segregation. And one of the candidates we turned to was the pathways that build embryonic polarity, the PCP pathway, which the major ligand of the pathway is the Wind11, and is actually selectively expressed in the margin. And Camila, what she did was to perform the same analysis in a mutant embryo that lacks Wind11, so it should lack embryonic polarity. And indeed, she immediately observed that these networks are not polarized anymore. So the giant cluster does not exhibit this strong geometrical bias, and no material boundary is visible along the animal-vegetal axis. Now we've performed the same analysis for the contact angles and the cell-cell tension. We observe now a flat distribution. So the cell-cell tension is now isotropic and no gradient is observed. So how is the segregation in these isotropic cellular networks? So first, the marginal cells for the Wind11 mutant embryos, they are competent, so they do become specified. However, if we look at in close up to the zone, we see that we have an irregular segregation. We see positive cells that you see here in the blue curve, faded cells so the specification happens a bit further from the margin, and not all cells close to the margin are becoming positive. And in this case, so we have a giant cluster that is not sharply defined at the end of the marginal area, and the specification area also does not show a sharp boundary. And this is actually kind of where we stand up with this project. So I will quickly wrap up of what I have talked to you today. So I have shown you evidence about that the zebrafish blastoderm undergoes a temporal rigidity percolation phase transition, which relies on cell connectivity and cell-cell adhesion. That there is also a spatial rigidity pattern in the blastoderm that coincides with the spatial fade pattern. A cell-cell adhesion gradient sets a sharp material boundary between the germ layers, and in its absence, the material boundary is perturbed and the segregation is imprecise. And now we're focusing to explore what is the mechanistic link between the material properties and the morphogen signaling, and we hope we will know more soon about this. So I would like to thank quite a few people, as I mentioned, the Heisenberg group, my collaborators, Bernat and Eduard, and all these people that they started in the new lab with me, and especially Camila that she took over this project. And thank you for your attention.
Fantastic. Thank you so much. I've been following this work and for a while now and really, really enjoying it. One question that I want to start out with for me is just thinking about, you know, what stands out to me before you talk about the network, showing the networks, is just the density difference. And so if you were to just look at these same plots and just as a function of density, would you see the same sharpness, or do you really need to look at percolation networks in order to see exactly where that sharp phase transition is?
Yeah, so density correlates until a point. So of course, if we have two dense tissues, they will be very connected. It will have much less denser tissues, they will be poorly connected. So it will agree with viscosity, but when we have in the middle, it seems that density is not sufficient. So in the wild types, we found that density can also, so cell fraction can also explain, but when we do the experimental manipulations, it seems that it's the adhesion and the connectivity that is important, because we have created mutants that they have less interstitial space, and we see that the fluidization still happens, but there the adhesion is more defective. So we think that it's not a passive density, but it's very hard to decouple.
Thanks. Fantastic.
Thank you. I'll take the first question from the audience. This question is from Abhishek. Are spatial rheological differences dependent on needle penetration depth?
Good question. We don't think so, although we haven't, let's say, tried to penetrate more, but we have mutants that the marginal tissue is fluidized, and then we can detect it. So especially we have created some transplantation assays where we put some fluidized central tissue to the margin, and there if we aspirate this tissue that is on the margin is still fluidized. So technically we think it's very accurate.
Right, great. From Seth Donahue, he says, great talk. Regarding the gradient of rigidity at the margin of the embryo, he wonders how much of this gradient would be expected to result just from proximity to a physical boundary, independent of, say, patterns of gene expression. Or put differently, can we assume that the cells are jostling around in space and connecting when they contact each other, and that a cell can't jostle through the boundary at the margin?
Yeah, great question. I had more data, but not so much time, but I can answer these two questions. So the first one, we also thought about it, but what we did was to block specification. So we have a nodal receptor mutant, and what we see is that the rigidity there is also messed up. So sometimes the giant cluster is unpolarized and it's not always on the, so it's not, so we think that the initial anisotropy is set by the morphogen per se. So even cells that are not fated, the anisotropy changes. Now on the second part, to address this, what we did is to go, because the CPOC signal is the downstream gene, so this is a very logical explanation that the cells may become correctly specified, but they just go around. So what we did is to look at very early markers of the specification. And this is, for example, the localization of SMAT2, which is downstream of nodal. So this is like an early marker that the moment the cell recognizes the ligand for specification, this marker goes to the nucleus. And there we can see that we have defects early on. So we still don't know if it's on the morphogen diffusion, the phenotype that stems for, but at least it's not because of cell motility that it comes after the specification.
Thank you so much. Next question is from Esteban Huichman. Says, super interesting talk and new results. Is it possible that planar cell polarity affects signaling in addition to connectivity?
Yeah. So of course we have the PCP mutant. It was great to start with, but now we want to address this in experimental ways that will specifically affect connectivity. So for this, we started some experiments where we locally change connectivity and induce gradient through optogenetics, but it needs a lot of time to, so I cannot answer this question yet. Yeah.
Great. We have a very difficult question, I think, from Roberto Mayor, who asks, how do you distinguish between material properties having a role on cell specification versus the fact that different cells could have different material properties? More, how do you think about cause and effect? What is cause and what is effect?
Okay, so so if I got the question, is it a matter of scale here? So it's the cell having a different material property versus the tissue. So if this is the case, so what we have checked actually is that if you look at the whole field of view and you average the adhesion, so not in this ROI.
Maybe I can explain the question better. So first, very nice talk. So the question is, I thought at some point you implied that these different properties of the material could have an effect on the specification, for example, of the endodermal cells. But my question is, maybe these different cells have different material properties because the cells are different. Because some cells are endodermal cells and other cells are not endodermal and they have different properties. So that is my question. I mean, what do you think is the cause and what is the effect of?
Yeah. So I don't have the answer yet, but we tend to think that it's more on a tissue scale property, because what we see is that if we average, let's say we didn't measure cell viscoelasticity, but if we average the adhesive properties of the whole field of view between the wild types and the Wind11 mutants, they are on average the same. They also have the same connectivity, but it's the polarity that changes. So, however, this doesn't mean that the cell viscoelasticity could still be different. It's just on different locations. So yeah, this we have to check. And thanks for the comment. Yeah.
Okay, so the next question is from Lucas, says, thank you for the talk. And does SLB mutant affect levels of ecadherin and mesendoderm, and therefore the giant cluster formation?
Yeah. So about levels, we don't know, but it's possible that affects the organization of the ecadherin. And this is something we have to look closer. There are several lines of evidence that suggest that PCP affects adherent structure formation, either through endocytosis, either through structure of the contact. So this is something that we will need to check.
Yeah, I had a related question, if I can. Do you think there's also contractility in play, because often it's a balance between the contractility and the adhesion, right?
Yeah. So definitely, because when we affect the other, the other gets affected.
But is it known in these embryos whether there is differences in the levels of contractility in, endoderm versus?
Exactly. So it is known in isolation. And but that's a problem, that once you isolate the cell and you expose it to media, that means the cortical tension immediately changes. And this is our biggest thing that we need to troubleshoot. Now, we hope that, so we have some optogenetic tools that they act either only on the contacts or only on the cortex. And we hope that we could decouple, but I think it's not so easy.
Yeah, for sure. I mean, it affects the endocytosis of adhesions and stuff like that. Yeah. I think we are in the end. If I'll check YouTube quickly. Thank you so much, both Kolade and Nicoletta, for giving wonderful talks. And this is our last session of the year, but we plan to come back next year. So we hope the enthusiasm.
We're already putting together a really great session for next year. So stay tuned, everyone, for that. But I think this was a really great way to finish off the year. Sorry, Ankita, to interrupt there. I just wanted to express my excitement and say also thank you so much, Nicoletta and Kolade. That was really fantastic from both of you.
And a happy new year to everybody.
Let's have a good year.
All right, I'll end the recording here. So bye to everyone on YouTube.